Good morning, everyone, and welcome to today's AAPS eChalk Talk. Today's eChalk Talk is hosted by Celerion. You may submit your questions at any time by entering them into the Q&A box of the Zoom platform. It is my pleasure to introduce our moderator, Joe Goodman, who is VP of Bioanalytical Services at Celerion. Joe, the floor is yours.
Thank you very much. It's my pleasure today to introduce my colleague, Joe Stanter, who will be your presenter today. Joe is the Global Director of the Celerion Molecular and Cellular Labs in Lincoln, Nebraska, and Zurich, Switzerland, where he's leading the GLP and GCP compliant testing to support the development of new modalities such as cell and gene therapies. Prior to joining Celerion, Joe was at Freeline Therapeutics as their Director of Bioanalysis focused on the bioanalytical support for gene therapy trials and companion diagnostic, analytical and clinical development. He also brings a wealth of bioanalytical experience for small and large molecule modalities, biomarkers, and clinical and non-clinical development through his senior roles at LabCorp and Hammersmith and Medicines Research. And Joe has also been involved in non-profit industry consortia such as European Bioanalysis Forum, discussing cell and gene therapies with various teams. His presentation today showcases an AAV shedding assay to support gene therapy clinical trials, where he will incorporate insights and discussions from the recent AAPS NBC conference in San Francisco. So with that, Joe, I will hand over to you for your presentation.
Great. Thank you so much. Thank you, Joe, for the kind introduction. Everyone here, I can see the numbers are going up. So thanks everyone for joining as well to this talk. And I will progress the slides forward. So I've got also prepared a session description, objectives and overview. But I think because everyone here has already joined, I think hopefully that's done a good job to attract as many as possible to join listen to this webinar, which is very close to my heart. There's also a quick overview about my bio, but Joe has done a fantastic job already to introduce me, so I'll skip that as well in the interest of time, and that hopefully will leave us more time as well in the end to ask some questions and hopefully engage in a very good discussion as well. So with that, I will get started with shedding and maybe a little introduction as well on why this is I think such an important topic, and then a bit of the regulatory background as well on why this has come especially into our bioanalytical and regulated bioanalytical laboratories as a topic and as an issue for us to tackle and to address. So shedding is in particular for a gene therapy product. It means that the gene therapy drug is then also found in the excreta and the secreta, also through the skin potentially as well of gene therapy products. So there's guidelines out there from the EMA and from the FDA as well that say during the clinical study, in particular, the patients, the subjects, the volunteers need to be monitored for shedding as well, because the shedding of the product needs to be understood. Now, the guidelines that have actually been written for this have been written a long time ago. So the EMA came out in 2009, and then afterwards the FDA one. So when you read through them as well, I feel like also they're a bit outdated now, especially with regards to what we already know about shedding, and also with the vectors that we see coming through. AAV has established itself as a very, very good platform that many studies we see at the moment in the clinic going through, and also some marketed products, absolutely fantastic products that make a real life to patients. So that's why I feel like also I want to address this here as well, and also from the feedback from the NBC as well from last week, that maybe we can take this into our companies as well, and hopefully influence a little bit as well on how the future clinical trial protocols are written, so that we don't just follow the guidelines for the guidelines' sake, but we really generate meaningful data that helps the progression of those studies through the clinic, and also makes the clinical trials as streamlined as possible. So if you look into detail for the FDA recommendation for shedding, it is very, very detailed in terms of how the clinical trial protocol should be written and what should be included, even down to suggested sampling from day 1, 3, 7, 10, and then thereafter weekly testing. So, this is obviously, it is only suggested, but we do see that in very many protocols being incorporated as a given, and if you then multiply that with the various different matrices that you'll find, usually 5 or 6 different matrices, plus very often blood as well, which is not shedding matrix, but it's very often included in the same assay. You end up with a lot of sampling, with a lot of samples, and a lot of inconvenience for the patients as well, and also a lot of recall of those patients into the clinical trial unit to provide those samples. And then once the shedding is, well, if you've got 3 consecutive negative results, the shedding is finished, per se, and the sampling can stop. So again, that information needs to go back to the clinic that has administered those patients, so that the sampling doesn't need to take anymore. So again, that is something that requires quite unique communication paths, turnaround times, and something that therefore really has a unique context of use within our bioanalytical world, to make sure we can comply with the regulations, with the protocol, with GCP, and make sure, well, the trial and the patients go on as fast as possible. So one of the key aspects as well that I mentioned in the EMA guideline as well is it actually mentions qPCR, and this is sort of, I guess, shows also how outdated it is. It doesn't mention digital PCR, for example, but it mentions qPCR in there, and it has to be a sensitive and reproducible method, and that's why it's sort of preferred. Also, maybe I go back to the FDA ones. It does say that the shedding assay is not expected to be validated, but the assay should be qualified and meet the minimum performance capabilities. And that's just how it stands in the guideline. Now, for us as bioanalysts, like, this is just like, it opens so many questions. And these questions, I've thought about, and hopefully I try to address here in this study as well. So one, when we sort of dissect how to analyze a sample, any sample, and the shedding samples in particular, we can separate it out into sort of 5 different steps. And really, from my point of view, all of those steps are really equally important to generate robust and reliable data, from the sample collection device to how the sample is handled, and then obviously DNA extracted, and then how the DNA is evaluated, and then also how the data is reported. All of those steps, hugely important. We can have a webinar on each one of those steps easily. And when you look into the sort of white papers and guidelines and sort of people in the molecular space, you'll soon realize that most people talk about the validation of a PCR method, and it's really focusing on the validation of just the qPCR part of the method, and that's sort of captured in many of the white papers and publications and papers out there in our field, that sort of focus on that aspect really, and that sort of that really got me thinking, because from my point of view, the DNA extraction is really just as important to generate accurate and precise results. So that's why, I'll show here as well how we've approached it, and also that we spike standards and QCs before we do the extraction, which from a general sort of feedback point of view as well is quite unusual in the molecular laboratory space. But we've got some great data, and we show that it works, and it can work, and it can work very, very nicely. So for this, and to be allowed to show you some of the data, we purchased this AAV8. It's just a GFP a protein in there in the plasmid, but it's commercially available, so it comes with everything that we expect in the biological world, a C of A, for example. So we can start working on this and generate some of our own data. So for us, the validation, to call something validated, we would set up predefined parameters with associated criteria, so the acceptance criteria. Think about the context of use. So I'd say tick, we've done that now. We've looked at why we need it and the regulation behind it. And then so the next steps would go into in assay development, and then in validation. As long as we've got a validation plan, we execute the validation according to the plan and have the acceptance criteria, and then write the validation report. The assay is validated for all intents and purposes. So again, sort of if you think back about the FDA and what they've written in their guideline, that the assay doesn't need to be validated and it needs to be qualified. Again, it's such a gray area that for me just sits very uncomfortably, especially when we're working with real clinical trial data and real patient data that has a lot of effort behind them, and it's hugely important, and our duty really to produce the best possible data from that. So one of the actual guide, apart from white papers, the actual guideline though that I quite like is from the CLSI, which is written for diagnostic labs, but I do like the way how it's approached. Keeps it flexible, gives you a lot of tools in there on how to approach it and how to think about it. And so this, we use that as a sort of guiding principle behind it to, well, first of all, set up our lab, but also, in our lab, we follow many of the CLSI guidelines and recommendations on how to do molecular methods, but also this particular one for a quantitative molecular method, and develop the assay accordingly. So we start off non-controversial with the design of our primers and probes. 5 primers and probes is a good set. We give every scientist in our lab usually the chance to design some primers and probes, and then each and every one of them gets ordered. They're very cheap to do, to order and get, and then they'll all be put to the test against usually 3, maybe 4 master mixes that we have available as well. Because experimentally, the primers might be really, really nice to design, but experimentally, that's really, really, really where it shows what the difference are. So then we go to the qPCR optimization. So that is what most papers are about, and this is really about the instrumental optimization. Define obviously the best primer and probe pair, what the LLQ is, what the LOD is, and the collinearity as well with the plasmid and the viral DNA. So you can measure reliably and have a reference standard to use as well. So this, I felt also with working with many labs as someone who outsourced assays to them, this is very often where the work stops for many assays. But it really, for us, we thought we make it our mission, and this is really where it starts. So LOD, for example, as well, is with regards to the context of use of a shedding assay, is one of the most important and the key parameters that need to be met. We can use several replicates, and you're really just looking for positive or negative response there. So you want to see how reliably can you still detect it, and you want to detect it with a 95% confidence if the sample is still positive or negative. So you spike in various different copy numbers, and here we worked on that quite a bit, and we've managed to achieve an LOD of 5 copies per reaction, which is one of the lowest you can possibly get. I mean, theoretically you could 3, but again, that's 5. We were very, very happy with 5 copies per reaction for this super sensitive assay. Now the next one is then the DNA extraction. And again, this is something where we thought, well, we really, let's take a step back and think, okay, what will be the best way to look at an extraction process, and also a protocol that we can use going forward to come up with the best possible extraction aspect. Because only if the extraction is really, really good, then it makes sense to have the qPCR makes sense, because only what goes into the qPCR will be measured. So therefore, we bought several kits that are available, also with the kit insert labels to say what they're suitable for. To my surprise, many labs that I used to work with, they would just take the kit insert and say, like, well, it says in here it's good for blood, so that's why it's fine. So, you know, we'll just do a few extractions and then that's it. Nothing we can modify anyway. There's nothing we can change. So therefore, you know, no need to think about it too much. And the acceptance criteria are very loose for people accept in the industry, normally between 20% and 120% recovery, which again, for me, was sort of my bioanalytical brain is just something that, like, from the very start has always irked me a little bit, because I don't think it's good enough. So that's why we put all those extraction kits to the test, with following the protocols and assessing them. And here I sort of just quickly go through it there. The main highlights, which is the ones that work particularly well for us, with recoveries in, you know, pretty much full recovery, 100%, 120, 92, 84%. Urine as well, this assay, the same kit that was good for all the other matrices actually didn't work very, very well for urine. So again, it really sort of confirms that all the kits really need to be tested individually against the exact reference standard that you're using. And in this particular case, our aim was to really, you know, get to a decent recovery, which is about 80%, which we would accept for all of our other bioanalytical assays, really. And we managed to achieve it here very nicely. But again, it needed a look of searching around for what best kit works, and it needs to be experimentally tested. Then you can also look at it in a slightly different way, where you say, well, actually, let's not look at it from just purely recovery point of view, because that's, like, you know, I think it's such a buy away thing to just want to get really good recoveries, but also look at it from a sensitivity point of view. And this is something to keep in mind with those kits, because some of those kits you can actually load a really decent amount of matrix onto them. So, for example, the Zymo kit, you can load far more onto the kit, while the recovery is only 37%. The actual sensitivity, the LOD in genomic copies per microliter, is actually much better than the QIAGEN kit, which had a very good recovery. And this is purely a volume difference in terms of how much you can load. Same is true for blood. If you purely go in for sensitivity, then the Zymo kit is actually better, even though the recovery is not as good, but you can load more. So definitely something to keep in mind, especially if you're trying to, like, push the sensitivity limits in various different matrices or biofluids. You know, same is true for biodistribution studies, of course. So it really makes a huge point on how much you're actually feeding into this assessment. So then I know what you're hearing. I know you're not saying it, but I can hear it, and I can see a few QAs as well, and hopefully I'm answering all the one of them were here already. It's like, well, I'm not surprised you can't detect anything in the feces or others, because you're using qPCR, and everyone has a huge rush to digital or digital droplet PCR, because it just overcomes all of that. So we put that to the test as well, of course. And the same recoveries that we had. So this is the Zymo kit, where we had no detection with qPCR. We actually get 4 or 5% on the digital one. So there is something in there, but again, nothing they would be happy with anyway. Same with the Promega kit as well. The inhibition that we see from that matrix, from the fecal stool samples, has totally wiped out the signal for our digital PCR. For our qPCR, and for the digital PCR, we do see recovery. But again, we're still below 20%. So, you know, nothing that really anyone would happy with. And the assays that work well. So our QIAGEN stool kit that we chose worked incredibly well. and it worked equally well on the digital as well as the qPCR. So really, if the qPCR is working, if the extraction is working, and if you've got your extraction kit set up properly, it will work on both. We've shown it worked equally well on both platforms that we had. So the qPCR, the digital PCR, is not going to pull you out of a bad performing extraction. We've found it really doesn't. So then the next aspect, once we've sort of selected the best extraction kit and worked that out, we then had a look at the final optimization. And this is for us, we bring everything together from the method development, each of the aspects that we've looked at separately, and find, well, how can we build the best possible bioanalytical assay? And for us, of course, that's spiking the standards and QCs before the extraction, because that will only that's for us the only way. I mean, we would do that for any assay that we do. Immunoprecipitation, LCMS assay, you name it, all of our labs spike into the matrix, and then we take it through the whole workflow. So we thought, oh, let's try that as well. And I'll show you the data for this. And this is also when you talk to people and how they've done it, and who's done it, and who's done it how, you come up with a huge variety of how people do it. And it's something that I found very interesting at the MBC, listening to the various different ways of how things are validated, and how the different approaches and how people do it in the lab. So I've tried to summarize them here in a few different ways. So replicates are a big thing, and single-kit is a new thing. So is our general perception in the industry. However, that's not always the case. So one approach is you have one sample, you do 3 extractions, and then you run 3 extractions onto your qPCR, and then you end up with 3 values. Surprisingly few people do this, but it is done out there. We've also found extraction done once and then run 3 times of the qPCR is a way of doing it. Again, probably not something I would want to do. But then now we start to get more interesting approaches where you've got one sample, you extract it 3 times, then you pool the extract, and then you run it 3 times on the qPCR. Again, interesting to see. I wouldn't even have thought about this. And then possibly as well, I haven't heard anyone mention this, but why not? Hey, if you've got 3, you get 3 aliquots, you pool them, you do 3 extractions, you pool them, and then you put them all onto the qPCR as well. So I haven't heard about that one yet, but I think it's only a matter of time. So anything is out there and anything is possible. But I think it's sort of the most standard way in many labs is to have one sample extracted 3 times. The recovery acceptance for many labs that I found, and many white papers out there as well, is between 20 and 120% of the recovery, which is really a broad spectrum. And for most methods out there, this is only assessed during the validation phase, and not with every batch or with every assessment. So this is done once, 3 different batches, 3 assessments during validation, and then tick recovery, sorted, pass. So then the standards and the QCs are spiked after the extractions and just under the qPCR. So you've got your standard curve and your comparison there. So that's I think seems to be a pretty acceptable way to do it. Now, the sort of, when I sort of think about it a bit more, then sort of do you end up with results, or 3 results, or average results, and you've got a CV value as well, which is quite nice, gives you all the variability. But then do you multiply it with the recovery percentages as well to get a true value of how much is the sample? And I think with most people I talk to, the answer is no. So you get the results, and the results are sort of normalized against microgram DNA or something like that, or the input volume, but the recovery is very often is not included in the final calculation of the results. So we thought, well, let's take a step back and what's the ideal way of doing it? What's the best way of doing it to come up with accurate results? So we spike samples, QCs, standards. We start with them at the beginning of our workflow. We extract them. Our extraction efficiency is over 80%. But all the standards and all the QCs undergo at the various typical titration levels as well through the same process. And then we analyze them once on the qPCR, because we've seen our qPCR precision is great. It doesn't need to be corrected for with averaging 3 results that pretty much tell you very similar answers and don't it doesn't really add much more data if you analyze the same sample over and over again. So yeah, very much a singlet approach, but we're really confident in the results because we know what we spiked in at the top was extracted, was measured, and is really a reflection of how much we put in and assess against the calibration line. So this is what it looks like for us. Typical standard curve that we have. We start off with a very wide dynamic range. Again, it's a nice thing about qPCR, and the limit of quantification here we are at 40 genomic copies per microliter, and very nice linear line that we can use to calculate our results. Anything below the limit of quantification we would report as below the limit of quantification, but above the limit of detection, so it's still positive, and that's really what matters the most for a shedding assay. And the assay validation and acceptance criteria we put down as below 35% for the accuracy and precision, and a recovery of above 80%. The sensitivity, so we defined as genomic copies per microliters, or for fecal samples or tissues above 1.5 genomic copies per milligram. So this, again, there's no consensus really on how to define the sensitivity. Many results that I've seen out there multiply the value by 1,000, so it will be 1,000 genomic copies per milliliter. So again, that's sort of a convention, I guess, that'll be good to set as well, so everyone knows how good the results are. But this is for us easily achievable in our assays. We were well below that, and yeah, very pleased with the results. Selectivity, so 80% of the individuals need to be above 35% of nominal. So again, so we don't use the percent CV or precision aspects too much here, because we know how much we're spiking into the sample, so we can use nominal value to back calculate, and again that gives us the biggest confidence in producing accurate results. For the specificity as well, so all the blanks is very simple criteria. All the blanks need to be below the limit of detection. So yeah, not too onerous for us to meet and not too complicated to do. Now, the sample analysis, the run design, again, this is something which is I think unusual, so that's why I thought I'll bring it to here as well, and hopefully can put it to the discussions as well. Yeah, we have 8 standards and a blank in singlet. The acceptance criteria are less or equal to 35% to the nominal value. Minimum of 6 standards have to be within the acceptance criteria. QCs, 2 singlet QCs are on the plate at the various different levels, 3 different levels, and then the blank needs to be below limit of detection, Otherwise, everything else needs to be within 35% and 4 QCs. So you can deactivate 2 QCs, 4 QCs need to be in, can't be on the same level as well. And then, yeah, sample is run once on each plate. So that then brings me to the sort of peculiarities of reporting results for the shedding, and I'll only touch on this very, very quickly. But this is something as well, a discussion from the MBC, which I think will be good for all of us to take to the project teams, is really how to schedule and time the collection of the samples. So in dosing day -1 obviously will be below the limit of detection. Then day 2, day 3, and then between dosing after a week of 1 to 11, the patterns look so similar. So there's a very quick ramp up of the copy numbers that you see. This is a feces sample I use as an example. So the results shoot up very, very quickly in all matrices up to week 11, week 12, so about 3 months in. And then after that, we start to get into the interesting bit, which is the decrease of the detectable DNA in the various different matrices, until we get below the limit of quantification, and then limit of detection, below limit of detection. As soon as we hit 3 below limit of detection, then no sample is collected anymore. But if there's only a week between each collection of the sample, and anyone who's done this assessment knows how difficult those assays are and how long they can take. So the turnaround time for such a result can easily be 2 to 3 weeks. By the time you've received the sample, by the time you've analyzed it and reported the results, 2 weeks can easily pass. Or if you're fast, okay, you can do it in a week. But again, by the time, have you missed the time to inform the clinic, to inform the next patient, to come in and collect a sample? And then what if you've collected a sample? I mean, you're gonna have to analyze it, right? Because otherwise, why, that this has been collected for no reason. And this is where it can get tricky as well, because very often the samples can hover around the limit of detection as well. You can have 2 negatives and then a positive again, or you can have 3 negatives, and then because you collected in a sample and you analyzed it, it's above the limit of detection. So this can go on for quite a while. So for that reason as well, it will be good, I think, in the clinical trial design as well, to especially remove this chunk in the first 3 weeks of sample collection. It really teaches us very, very little, and especially this is typical for an intravenous AAV. It's a very, very typical pattern that is repeated over and over again in the various different matrices, and I don't feel like we're learning anything, and it's very, very predictable what they're going to look like. The interesting bit starts at week 12. So I think this is where the sample analysis should really start. Interestingly, many protocols, this is where they start ramping down the collection of the samples, and then they go from weekly collection to monthly collections, and then you have to wait 3, 4, 5 months, 6 months before you can clear a patient. So if we think about this sort of aspect and maybe design the protocols with a bit more of that in mind, hopefully we can streamline the analysis and also make it a lot cheaper for everyone. It's an expensive assessment, it's an expensive assay, and the more streamlined we can do this, the more benefit it is for anyone. So yeah, on that note, come to the conclusion. So shedding assays have a unique context of use. Inhibition from different matrices, yeah, so of course possible. Different kits need to be evaluated and validated. Fast turnaround is needed to inform the next sample collection for the trial participants. And LOD optimization is really the main focus of the method development. It doesn't matter if the assay is really, really precise at the high levels. The LOD, that's where you're going to be generating the most results in your lab. So it is contributing to some sort of sample collection within the clinical trial protocol. I feel very strongly about it, that really this is not IVD. Please don't make it an IVD as a shedding assay. it will explode the costs and make it 10 times more onerous to do and deliver, and it's not worth it. I mean, the guideline doesn't even say it needs to be validated. So why should we take it to the next level and make an IVD? So best analytical practice is to spike drug product into the matrix before extraction. And we've shown here it really works nicely, and I think is the way to go. And the last one, adjust the clinical trial protocol to make sample collection only when needed, based on the context of use of the assay and what we already know from previous studies. So I would say collection every 2 to 3 weeks, and don't start too early. We're not learning anything more. And on that, I want to acknowledge especially everyone in the lab, Sylvia, Andrea, Petya, and Petra, and look forward to any of the questions that you have.
Thank you, Joe. We do have 2 questions. I think we've answered one, but maybe we can go just back for clarification in a minute. Yanshan was asking regarding the assay. You mentioned it doesn't need to be validated, but it should be qualified. There was a clarifying question around, does it need to be to GLP or general regulated bioanalysis standards?
Very good question. So it definitely does need to be to GLP, because this is particularly about the clinical study. So good documentation practice, yes, but GLP, no. I mean, there's no animals involved. GCP, yes, of course. Any sample that's being collected, of course, has to follow GCP. That needs to be informed us. You need to make sure in the lab, obviously, the informed consent form is reviewed, and it's in accordance with the protocol, and also how the protocol is written, and so on and so forth. So that definitely needs to be an aspect we need to consider. But other than that, no, it's a free-for-all. There's no guideline out there that tells you how to do it or how not to do it. So yeah, we can really let the best science and context of use guide us on how we approach this.
Thank you, Joe. And just a clarification, it was spiking the drug substance. There was a question asking whether it was the plasmid DNA or whether it was the drug product equivalent, i.e. encapsulated virus.
Yeah, very good question. And we did use the plasmid as well, very much so, linearized, of course, during the early development stages, especially when you want to look at your PCR optimization. Does it work? How does it work? How difficult? What's your efficiency, and so on and so forth. And also for something that you can use to get back to, if you want to do troubleshooting and want to spike into the various different extracts or matrices. However, the actual reference standard to generate the accurate results is the actual drug product. So is the encapsulated a drug product that's administered, and that's spiked straight into the matrix where you're trying to detect it from. So because the plasmid extraction is very different to encapsulated AAV extraction. So that's why for us it represented the best amount of a reflection of what the sample would potentially have in there. And the plasmid, you use different kits for plasmid recovery, and again, it's not something that was it's possible, but we didn't think it was a true reflection of what we're actually measuring.
Perfect, thank you, Joe. In terms of data and how data are used, there's a question saying, if we find, for example, that feces are positive for 6 weeks post-dose, what would be the impact on the program?
If we find that feces, I mean, it's absolutely possible. Yeah. I mean, I've seen data where feces have been collected even a year after the subject's been dosed. It does happen, and also in the guideline it does say, if you found sort of the bottom level of the shedding, then you can also stop collecting samples. Which is if it hovers around about the limit of detection, or if you detect very, very low levels as well for a long period of time, you can also stop. and yeah, I guess that's one of the aspects that need to be. That's why it's quite tricky as well to look at it, and how and scheduling the sample analysis as well. It's really good to look at the previous results that have been generated, and if you know there have been 2 previous results that are positive, well, you may as well wait until you have 3 samples in your lab and then run the next assay, because then you've got the best chance of actually measuring 3 negatives in a row. So again, that's how it needs to be approached. I mean, and this is also from a sort of a big collection of data, mainly of intravenously administered AAV types, which have a particular shedding pattern, and again, that's sort of something that's really really something we don't need to repeat in every single study. But then it really starts to get interesting in a sort of, you know, the long-term follow-up area, and that's why I sort of, the point that I'm trying to make is, like, the patient's already so stressed in the first 2-3 weeks, months of the treatment anyway. You don't need to additionally add, you know, loads more sample collections to that as well, when we know the data that we get from them is really, really minimal. Once it comes to 3 months, 4 months, 5 months, 6 months, that's when it starts to get interesting. And also it's interesting when you find some peculiar cases where suddenly, you know, the shedding results start to hover around the LOD, and you find it quite impossible to sort of, well, not impossible, but it's really difficult to make a clear-cut decision on what to do next. Collect another sample, stop collecting, you know, and obviously various different aspects that have on the logistics of that particular patient as well who had that treatment. So
great. I think we're getting close to time, Joe, and we'll just check on the questions. If my mouse would move, sorry. Are you, there's one last one that I think we can take before closing out. Are you subjecting controls to extraction prior to PCR? Do you prepare standard controls in each shedding matrix type?
Oh, yeah, yes. Yeah, yes and yes. So each matrix type is spiked with the standards and QCs. We usually treat all matrices in the same way. So we do for example, a batch extraction for semen, and we build a run plan together. We build the calcium QCs, and then they're all spiked into the individual matrix that we are extracting on that day, together with the samples. So freshly extracted standards, freshly extracted QCs, and obviously freshly extracted standards samples as well in that same batch. And then they're used to back calculate the results. I know where the question comes from, and I wish there was a simple answer, because we tried also, of course, to just extract the standards once and then freeze them, and then we can use them going forward just on the PCR and add them in there, and then everything is fine. It's not. It really is not fine, because once you add your QCs and spike your QCs into the matrix, and before the exit, and extract them on the day that you're doing the extraction, they don't match. So it really has to be done in one go, in one batch, just like we would do. It's just like we do for an LCMS assay, just like we do for an immunoprecipitation assay. You wouldn't even dream about doing them in a different batch or separately. You make sure they're separate, because it just gives you the best chance of having the best possible results.
Mm-hmm. Perfect. Thank you, Joe. And if there are any further questions, we can, of course, follow up offline. So
yeah, please do.
Yeah. Thank you. And I'll hand back to the AAPS team to close out the session.
Thank you so much, Johannes and Joe and everyone at Celerion, for this wonderful presentation. This presentation has been recorded and will be available on the AAPS website. Thank you again, and we look forward to seeing you at a future eChalk Talk. You may now disconnect from the webinar and have a wonderful day.